Good afternoon. My name is Jonathan Hall. I am a chemistry major at East Carolina University, and I was awarded the IRCA Mini for fall 2020. So today I'm going to be going over coping with copper and periplasmic FtrA. FtrA is a protein, an iron 2 plus transport protein that is inside of brucella. Brucella is an intracellular gram-negative bacteria that infects a wide range of mammals, including goats, pigs, cows, and dogs, and causes an infection in humans as well from those mammals. So humans can get it from consumption of raw meat or contact. Once it's inside of the human body, it affects things like the bone marrow, lymph nodes, spleen, and liver. And so, since it's intracellular, it needs to acquire everything from the host. And so, it's going to upregulate iron transport systems to acquire its iron. So, iron is very important for most living species, and it is the second most abundant metal. So, while it's really important, it's also toxic. Iron can react with water or oxygen to create superoxides and rust. Superoxides go through the body and destroy DNA, causing mutations and or cancer. That being said, in order to combat that, iron is always chelated or has a chaperone protein with it at all times to keep it from reacting with water or oxygen. In order to do that in brucella, it upregulates different iron transport systems. The three types of iron transport systems that it has is heme, iron 3 plus siderophore, and FtrA-BCD. Out of these three types, heme and FtrA-BCD are required for virulence. Specifically, this lab is talking about FtrA-BCD, which is a four-component iron uptake system composed of FtrA, B, C, and D. FtrA acquires iron 2 plus from the periplasm and transfers it to FtrB, which oxidizes iron 2 plus to iron 3 plus so it can travel down into the cytosol and be utilized by the cell. Specifically, we are talking about FtrA. So previous research in this laboratory has showed that FtrA binds to an iron mimic, which is manganese 2 plus, in a copper 2 plus dependent way. This means that in order for FtrA to do its proposed function, it must have copper bound to it. This was shown because manganese would not bind to FtrA without copper. However, over here on the right, when FtrA is bound to copper, manganese does bind. So here is the data stating copper binds at a 0.5 molar ratio, which means that each part of FtrA, because it's a dimer, is attached to two different coppers. So there's a histidine that is a bridge that connects to the other copper. All right, so what am I trying to answer? From the previous research, it showed in vitro that FtrA needs to be bound to copper in order to bind to an iron mimic. So I started wondering, since copper is also toxic, like iron is, and it always needs to be chaperoned, where is FtrA getting its copper from? So I looked at a metal that is biologically relevant, that is in the periplasm, free floating, and I found zinc. So I asked, does zinc bind to FtrA? And does zinc affect copper affinity or function of the protein? So once again, why do we care about zinc? Zinc is biologically relevant, meaning it is present in the periplasm of the bacteria. It's also present in the host cell. It mimics copper 2 plus, meaning the size to charge ratio is very similar. And as seen in the two figures below, the binding site between copper and zinc is very similar as well. So on the left, you have FtrA, the binding site for copper. And on the right, you have zinc copper SOD, and that is the binding site for zinc. Whenever it's under iron starvation, Brucella upregulates zinc uptake systems as well as iron uptake systems. So it's going to have plenty of zinc as well. Right? So in order to answer these questions, the first thing that we utilized was isothermal titration calorimetry, or just ITC. Specifically for this experiment, it's titrating metals into the protein of choice, which is FtrA. Once we did this with zinc to FtrA, we acquired different figures such as the KD, the delta G, the delta H, and delta S. And so this this just explains how it is binding to the protein. So what have I done so far? On the left, it is zinc 2 plus to FtrA, and then on the right, it is copper 2 plus to FtrA. So the copper 2 plus to FtrA was already previously done and has been published, whereas the zinc 2 plus to FtrA is the new information that is being compared to copper 2 plus. As can see from the table, zinc actually binds to FtrA 10 times more tightly than copper does. Also, whenever copper is titrated into FtrA zinc, a precipitate is formed, meaning that it is doing something to the protein. We just don't know what it is yet. So here are the graphs of copper being titrated into FtrA zinc. And as you can see, the mole-to-mole ratio differs a lot, so 0.21, 0.42, and 0.65, as well as a precipitate being formed. So it's really inconsistent, but zinc is obviously doing something to cause this difference in the copper binding. So in the future, we're going to be running zinc into copper FtrA to see if it can replace the copper. We're also going to be running zinc into different mutants of FtrA, specifically with the copper binding site, to see if it's binding at the copper binding site or another allosteric site. We're also going to be performing protein folding experiments such as CD and DSC to investigate structural changes in FtrA. And lastly, we're going to make isogenic FtrA-BCD. So we're going to take FtrA-BCD from Brucella, put it into E. coli, and test the effects of zinc in that. And then publish my work and hopefully graduate. I would like to say thank you to Dr. Banerjee and Amina for guiding and assisting me through this project. And then to the Department of Microbiology and Immunology, as well as the Department of Chemistry and undergraduate research at ECU. And here's our group. Yay.